Every empire that ever rose in the ancient world passed through this land. The Egyptians marched north across it. The Assyrians swept south. The Greeks renamed it. The Romans paved it. The Islamic caliphates absorbed it. 11,000 years of recorded history. Conquest layered on conquest, language layered on language, and yet the people living here today carry a genetic signal in their blood that points to a place none of those empires ever touched. A place not north, not east, not across the Mediterranean, south, deep south. Sub-Saharan Africa, more than 4,000 kilometers away, across an ocean of desert and savanna, with no recorded route between. The land is Jordan, and the DNA is still unexplained. To understand why the genetics are so strange, you have to understand how settled the story was supposed to be. Jordan sits at the heart of the southern Levant, the narrow corridor of land running between the Mediterranean coast and the Arabian desert, roughly where Israel, Palestine, Lebanon, and Syria cluster today. This is not ordinary geography. This is the ground where human civilization made several of its most consequential decisions. Around 12,000 years ago, Natufian hunter-gatherers built the earliest known semi-permanent settlements here, along river systems and hilltops that could support a small, mobile people beginning to experiment with staying in one place. 2,000 years after that, the Neolithic Revolution took root, literally. The world's first farming villages emerged from this soil, the first domesticated wheat, the first corralled animals, the first architecture built not for shelter from a single season, but for lifetimes. By the Bronze Age, roughly 3,500 to 1150 BCE, the southern Levant was organized into a loose network of city-states sharing a common material culture. Archaeologists call these people the Canaanites, though that name encompasses dozens of distinct communities. They traded, warred, intermarried, and built. They are the ancestors the historical record gives to the people of this region. And the genetics for a long time seem to confirm the story. In 2020, a landmark study published in the journal Cell, led by researchers at Harvard University and the Hebrew University of Jerusalem, analyzed genome-wide data from 93 ancient individuals excavated from 9 southern Levantine sites, with dates spanning from roughly 2400 BCE to 900 BCE. What they found was continuity. Deep, stubborn, remarkable continuity. Modern Jordanians, along with Lebanese, Israelis, and Palestinians, typically derive more than 50% of their ancestry from these Bronze Age populations. The Canaanites did not disappear. They became the substrate on which every later culture was built. A 2011 study published in Genetic Testing and Molecular Biomarkers added another layer of confirmation. Researchers tested 150 unrelated Jordanians using ALU insertion polymorphisms, genetic markers that trace deep population movement, and found that Jordanians were not significantly different from populations in the UAE or North Africa. The conclusions were direct. The original genetic profile of the Jordanian population, described as very likely Arabian Semitic, had not been subject to significant change despite the succession of several civilizations. The Nabateans rose and fell at Petra. The Romans annexed the kingdom in 106 CE and renamed it Arabia Petraea. The Islamic Caliphate swept through in the 7th century. The Ottomans governed for 4 centuries after that. Each wave left cultural and architectural sediment. None of them, according to the genetic models, fundamentally rewrote the population. It is a clean story, a coherent story, a story that makes sense of how a people could persist across 11 millennia of recorded upheaval. The DNA on the whole says, these people were here, they stayed here, they are still here. And then you look at one particular valley, and the story stops being clean. Here is where it gets complicated. In 2005, a team of researchers published a study in the Journal of Human Genetics. Flores, Macameyer, Laruga, Cabrera, Karadsha, and colleagues, titled Isolates in a Corridor of Migrations, a high-resolution analysis of Y chromosome variation in Jordan. The study used 46 binary markers to map the Y chromosome landscape of 2 Jordanian populations. One was from the metropolitan area of Amman, the capital. The other was from the Dead Sea Basin, a geographically isolated community living at the lowest point on the surface of the Earth, approximately 430 meters below sea level. The Amman sample was unremarkable. It looked genetically exactly like its Levantine neighbors. Ordinary. Expected. The Dead Sea sample was something else entirely. A Y chromosome haplogroup is, in the simplest terms, a lineage marker, a surname passed down not in writing but in the body itself, transmitted from father to son across hundreds of generations without alteration. The dominant haplogroup across all Middle Eastern populations, including Amman, just 60 kilometers away, is haplogroup J. It runs at roughly 50 to 70% across the region. In the Dead Sea sample, haplogroup J appeared at 9%. 9. The community was not merely an outlier, it was genetically invisible to the world surrounding it. What had replaced J? Haplogroup R1 star, defined by the marker M173, appeared at 40%. In Amman, R1 star runs at approximately 1 to 2%. In Turkey, 1%. In the broader Levant, essentially 0. The researchers searched their global databases for a comparable frequency. They found one match. Northern Cameroon, Sub-Saharan Africa. That alone should have been enough to stop the field. But then the mitochondrial data arrived. Mitochondrial DNA is the maternal inheritance, passed from mother to child, tracking lineage through the opposite line of descent. A 2008 study by Gonzalez, Karadsha, Macameyer, and colleagues, published in the American Journal of Human Biology, analyzed the mtDNA of the same Dead Sea community. Haplogroup U3, which in most Middle Eastern populations runs at negligible frequencies, appeared in the Dead Sea sample at 39%. The highest U3 frequency recorded anywhere in the Middle East, higher than Iraqi Jews, where it runs at 30%, higher than Romani populations, where it sits around 10%. Two separate inheritance systems, two separate biological clocks, two completely independent lines of evidence, both pointing to the same anomalous signal in the same isolated community at the edge of the same impossible valley. Not a rounding error, not contamination. Both the paternal and maternal lineages of the Dead Sea community mirror each other in their departure from the regional norm. The researchers' own language is careful but clear. This community clearly behaved as a genetic outlier in the region, and its mtDNA lineage composition mirrors, in geographic origin and haplogroup frequencies, its Y chromosome pool. When both independent genetic clocks agree on the same wrong answer, the answer stops being wrong. It starts being the question. The first theory is the one scientists reach for first because it requires no dramatic revision of history. The Dead Sea Basin is one of the most extreme environments on Earth. Its surface lies 430 meters below global sea level, lower than any other point on dry land. The terrain surrounding it is a maze of salt flats, eroded wadis, and cliffs that rise abruptly on both the eastern and western shores, creating natural barriers that effectively seal the basin from its surroundings. A small founding population settling here thousands of years ago would have found themselves within a few generations functionally isolated, cut off from the migrations and admixture events continuously reshaping the genetics of neighboring populations. Genetic drift works like compound interest, applied to randomness. A founding group of fewer than 200 individuals, practicing endogamous marriage, marrying within the community, across 50 or more generations, can theoretically shift haplogroup frequencies by 30 to 40 percentage points. The mathematics are well established. The Flores 2005 paper itself invokes exactly this mechanism. Isolation, strong drift, and slash or founder effects are named as the primary explanation. It is a reasonable hypothesis. It has happened elsewhere. Haplogroup amplification through founder effect has been documented in island communities, mountain valley populations, and desert oases across the world. The physics of small populations in enclosed geographies are well understood. But here is the limit. Founder effect explains the magnitude of what happened in the Dead Sea Basin. It does not explain the identity. Drift amplifies whatever was already present in the founding population. It does not manufacture a new haplogroup from neighboring frequencies. It cannot explain why the specific marker that drifted to 40%, R1 star M173, is the same one clustered in northern Cameroon. Somewhere, at some point, there was a founder who carried this signal. Founder effect explains what happened to that signal after the founder arrived. It says nothing about where the founder came from. The second theory is more complicated, and it requires a different kind of imagination. What if R1 star M173 is not an anomaly, but an archive? Not a signal imported into the Jordan Rift Valley from somewhere else, but a signal that was there first, a surviving remnant of a population layer so ancient that the Neolithic Revolution, the Bronze Age, the entire succession of civilizations we call history, passed over it without erasing it. Haplogroup R, the ancestral clade that contains R1 star, has been dated to approximately 25,000 to 30,000 years ago. This is not the Bronze Age. This is not the Neolithic. This predates farming by 15,000 years. It predates the Canaanites by 22,000 years. Under this theory, the Dead Sea community is not genetically strange, it is genetically primordial. A relic population, sheltered in an extreme environment, carrying a haplogroup that was once widespread across the Afro-Eurasian corridor before successive waves of Neolithic farmers and Bronze Age pastoralists diluted or replaced it everywhere except the most inaccessible refugia. The 2020 Cell study by Harney and colleagues gives this theory indirect support. Even the Canaanite Bronze Age populations of the southern Levant were not a single ancient people. They were themselves mixtures of earlier Neolithic Levantine farmers and migrants from the Zagros Mountains and Caucasus region, with traces of ancestry from sources the study acknowledges it cannot fully model. Jordan's genetic history contains at least 3 distinct ancient layers before any Arab migration. The possibility of a fourth, deeper, older, pre-Neolithic, is not implausible on its face. Researchers are careful to frame this as probability, not certainty. The dating of haplogroup R's origin is contested. Its dispersal pattern before the Neolithic is poorly mapped. The absence of ancient DNA from the Dead Sea Basin itself means this theory cannot yet be tested directly against the archaeological record. It remains a coherent possibility, not proven, not disproven. But this theory too hits a wall. If R1 star was once broadly distributed across the ancient world, why does it survive at high frequencies in only 2 specific geographically separated locations, the Jordan Rift Valley and northern Cameroon, with essentially nothing in between? Relic populations in refugia should appear in clusters across the former range of the haplogroup. The pattern in the data is not a scatter, it is 2 points. That specificity demands an explanation the relic theory does not fully supply. This is the part the records don't explain. The third theory, and the one most researchers are hesitant to say out loud, suggests that what we are looking at in the Dead Sea Basin is not a relic of the ancient past, but a trace of a migration that history never recorded. The argument runs as follows. At some point within the last 2,000 to 5,000 years, well within the time frame of documented human activity in the region, a population from sub-Saharan Africa carrying high frequencies of R1 star M173 moved northward through the East African corridor, through the Horn of Africa, and into the southern Levant via the Arabian Peninsula. Their genetic signal, carried by a relatively small number of individuals, was absorbed into the Dead Sea community and subsequently locked in by that community's geographic isolation and endogamy. What makes this uncomfortable to dismiss is not R1 star alone. It is the second data point from the same community. The Flores 2005 paper also recorded haplogroup E3B3A-M34 at 31% in the Dead Sea sample, a frequency the authors describe as only comparable to that found in Ethiopians. 2 African-linked haplogroups, same community, same valley, same direction. The data doesn't confirm this theory, it only fails to rule it out. The Nabatean kingdom, centered at Petra and at its height from roughly the 2nd century BCE to its Roman annexation in 106 CE, controlled the incense trade routes connecting the Arabian Peninsula to the Mediterranean. Those routes ran through sub-Saharan and East African ports. Traders, soldiers, enslaved persons, small numbers of people moving along established corridors over centuries, can deposit a genetic signal that later isolation amplifies into something that looks, in retrospect, like a population event. The Nabateans did not need to orchestrate a migration. They only needed to keep the roads open. You are standing at the edge of the Dead Sea before dawn, the Jordanian shoreline. The air is heavy, salt thick, chemical with brine and the faint undertone of sulfur from the mineral-dense water just below you. The ground beneath your feet is not quite solid. Salt crystals have crusted the shoreline into a brittle, shifting surface that crunches and gives with each step. Nothing lives in this water. Nothing has ever lived in it. The salinity is 9 times the ocean. The surface lies 430 meters below the world you came from, the lowest point on Earth, and standing at its edge feels geometrically wrong, as if the horizon has been pulled too close by the weight of all that descent. The cliffs of the Judean desert glow on the far western shore, catching the first gray suggestion of light. They are not distant. The Dead Sea is narrow, but the silence between you and them is absolute. People have lived near this water for a very long time. Not because it is hospitable, it is not, but because the freshwater springs at the basin's edges and the trade routes threading through the surrounding wadis made survival possible for those willing to stay when others moved on. They stayed. Through Egyptian expansion, through Assyrian collapse, through the rise and fall of the Nabateans, through Roman administration, through Islamic conquest. They stayed when the empires left. They stayed when the caravans stopped running. In 2005, a team of geneticists collected their DNA and ran it through haplogroup analysis. What came back did not match the population 60 kilometers north. It did not match anything in the Middle East. It pointed, quietly and insistently, to a world 4,000 kilometers south. What does it mean to carry a lineage that has no genetic neighbors? What does it mean that your Y chromosome points somewhere else entirely, somewhere beyond the Nile, beyond the Sahara, across every boundary that history drew between you and that distant place, while you have been standing in this valley, beside this impossible water, longer than anyone can remember? Here is the twist that recontextualizes everything. Jordan was supposed to be a crossroads population, blended smooth by 10,000 years of migration, conquest, and trade, the way that high-traffic corridors usually are. The expectation in population genetics is that crossroads populations converge. The more people pass through a place, the more the local gene pool reflects the average of everyone who ever stopped. Jordan does not converge. Jordan accumulates. The extreme environments within the country, the Dead Sea Basin, the Wadi Rum Desert, the remote highland plateaus, did not simply isolate their inhabitants from the crossroads traffic above them, they preserved them. Every empire that swept through the Levantine corridor marched along the high roads, the trade routes, the navigable paths between coasts. The communities in the basin below sea level, in the canyons cut too deep for armies to bother with, in the desert formations too remote for administrators to administer, they were passed over. And in being passed over, they became archives. The most extreme environment became the most faithful record. The 2019 mitochondrial DNA study, published in a major genetics journal, tested 293 unrelated Jordanians, Arab Jordanians, Circassian migrants descended from Caucasian refugees who arrived approximately 140 years ago following the Russian-Circassian War, and Chechen migrants from the same era. The researchers identified 102 distinct haplogroups across the 3 communities. Only 6 were shared. 6 out of 102. Jordan is not one genetic population wearing a single national identity. It is multiple ancient populations, each carrying their own private archive, living alongside each other for generations without merging, because the geography and the history and the endogamy of each community conspired to keep the records separate. The Circassians did not absorb into the Arab population. Adapted. The Chechens maintained their own lineage. Adapted. The Dead Sea community preserved its ancient signal through every conquest. Adapted. What the title calls not Arab, not Canaanite, is not a gap in Jordanian identity. It is an excess of identity. Jordan's DNA is not a puzzle because something is missing. It is a puzzle because too much survived. And the modern frameworks for classifying ancestry were not built to hold that much complexity in one small country. Jordan's DNA is not a local anomaly. It is an argument about what happens to human genetic history when geography refuses to cooperate with the stories we tell about it. Population geneticists have spent decades discovering that the populations we call indigenous to any given place are rarely the first people there. That every landscape, no matter how long settled it appears, has been overwritten and re-overwritten by migrations that left no record in language or architecture or memory. But the inverse is also true, and Jordan is its demonstration. The most extreme environments, the places that empire and agriculture and commerce found least useful, became the vaults where those overwritten layers survived. The Dead Sea Basin and northern Cameroon share a haplogroup, not because these two communities were ever in contact, but because both of them, in their respective extremities, one below sea level in a salt basin, one in the mountain ranges of Central Africa, were bypassed by the same Neolithic flood that replaced the older world almost everywhere else. They are two surviving pages from the same erased manuscript. Every time a Jordanian today submits a sample to a commercial ancestry database, the algorithm assigns them to broad Middle Eastern categories. For most Jordanians, that result is approximately correct. For the communities along the Dead Sea, researchers are careful to note, the algorithm may be pointing at the wrong continent entirely. The full history of who built this land, who survived in it, who was preserved by its hostility, that history is still being excavated from the genome, one haplogroup at a time. Not Arab, not Canaanite. The question of who the Jordanians truly are has not been answered. It has only, finally, been asked correctly.